So we always, at this point, say, welcome to Iowa State University of Science and Technology. Right. I have to say, Jonathan and Laura worked Friday, got the whole system working flawlessly, and so the best laid plans. Anyway, welcome, and thank you very much for coming to tonight's lecture, the Fall 2011 College of Liberal Arts and Sciences Dean Lecture Series. I'm David Oliver. I'm the interim dean of the College of Liberal Arts and Sciences. And it's my pleasure to host this very special event. Tonight's lecture is the 11th Dean's Lectures in the series. LAS is dedicated to excellence in faculty research, scholarship, and discovery, and this lecture series highlights some of our very best efforts. Twice a year, we invite our top academic researchers to present lectures on their own varied areas of expertise. These lectures stimulate intellectual discussion amongst the faculty, staff, students, and the greater Iowa State community. I want to thank the ISU Lecture Program for their role in helping sponsor this event. As always, they do a wonderful job. I knew someone was going to laugh at me when I said that, right? Tonight's lecturer is Jonathan Wendell. Jonathan joins a distinguished list of LAS faculty members who've been part of this series, which was inaugurated by my predecessor, Mike Whiteford, in 2006. These researchers and scholars have shared their knowledge on a wide range of topics, ranging from woody bamboo plants to the physics of a curveball, from West African primates to dead poets and stolen poetry, from Old World Mediterranean antiquities to New World Mediterranean cruises to tomorrow's futuristic nanotechnology. And from the humor of Putin's Russia, a very humorous subject, we were told, to elegant fine details of protein structure. The depth and breadth of our speakers mirrors the academic diversity found in the College of Liberal Arts and Sciences. Tonight's lecture, I assure you, will be equally informative and enlightening as our previous presentations. Jonathan Wendell will present a lecture titled Genes, Genes, and Genomes. You know, that's a visual joke. It really is sort of unfair setting it up to us. Genes as in blue, genes as in little DNA sequences on chromosomes, and genomes. Exploring the mysteries of cotton. He is chair of the Department of Ecology, Evolution, and Organismal Botany, Biology. Wow. Okay, you can see why I'm an interim, right? After that, I'm going to lose my job. Ecology, Evolution, and Organismal Biology. He's an internationally feted plant biological researcher, a wildly popular teacher, a 29-state marathoner, and as I understand, 49-state marathoner with Arkansas still to go, and so that will be conquered. Editor of several premier science journals, a wine connoisseur, and the owner-operator of the largest cotton plantation in the state of Iowa. He's the person who's brought together the world's largest collection of wild cotton species and the principal investigator for the NSF Cotton Genome Project. For those of you that aren't aware, no, cotton is neither a variety of corn or soybeans. And so people always ask us, why do you study cotton in the state of Iowa? And so what Jonathan will show you is that cotton is a marvelous model organism for studying a very fundamental question of evolutionary science. We all know enough about Darwin's evolutionary hypotheses and theories to understand how they talk about how a species changes with time. But there was a fundamental question that's been present in biology for hundreds of years, and that's the source of new species. Where do new species come from? And Jonathan's work on cotton is one of the seminal experimental works to really detail where new species come from. And that's part of the story we're going to hear today. Jonathan Wendell earned a PhD from the University of North Carolina in 1983. He's a fellow of the American Association for the Advancement of Science. Please welcome Jonathan to the stage as the speaker of the 2011 Dean's Lecture Series for the College of Liberal Arts and Sciences.
Wow. Well, thank you, David, for that delightful, informative, and in some respects embarrassing introduction. Quite sincerely, I'm delighted for the invitation. Feel very honored. I'm flattered to be here tonight. I'd like to thank all of you for coming out on this dreary evening to hear what I have to say about this marvelous golden era in biology that we all find ourselves in and why it's such a fantastic time to be an evolutionary biologist. A nice place to start with any talk in evolutionary biology is with this fella, Charles Darwin, for whom we just celebrated a bicentennial birthday filled with all kinds of conferences and talks, and the sesquicentennial of the publication of his book that helped change the world. And these milestones, like milestones in our own lives, give us opportunity to reflect on the past and where we've come, and in the context of tonight's talk, to consider the impact of some of the really breathtaking technologies that we so enjoy today on questions that were fundamentally important to Darwin in his time, such as how does speciation occur, as David Oliver could have given the rest of my talk, I think. How does the evolutionary process lead to adaptation and the origin of new structures, and what is the nature of variation that natural selection acts upon? These are central questions in evolutionary biology. They're challenging questions. They're difficult to answer. They aged Darwin considerably. As much as Darwin liked finches, he liked sheep and oats and barley a lot more. Why? Because he understood that evolution takes place over huge amounts of time, large amounts of time that are difficult for humans to conceive of, but that humans also practice evolution, if you will, natural selection, in a very intense way in their own farms and fields and backyards. And so he astutely recognized this. He wrote books on the subject, and he said, "It seemed to me probable that a careful study of cultivated plants would offer the best chance of making out this obscure problem. I may venture to express my conviction of the high values of such studies, that is, studies of cultivated animals and plants." And so tonight I'd like to tell you a little bit about this for the cotton genus, shown here in its, for those of you who are from Iowa and haven't left, shown here in its highly annualized modern form. There's nothing like this in nature. This is a product of human domestication. It's a product that we're all very familiar with. I show here the school uniform of the Iowa State University undergrad. And actually, for the rest of us, if you pay attention to statistics about how many genes we all own and how much in love we are with our own genes, so cotton is extremely important in our own lives. Probably everyone in the audience is wearing cotton on some part of their body. Don't tell me where. It's not just jeans, but all kinds of clothing articles, all kinds of other consumables that you find in the grocery store come from cotton. Not just the cotton fiber, which, by the way, and you can practice saying this, are single-celled epidermal trichomes on the seed, but also from the seed itself, which is a valuable source of oil and meal seed for animal feed. They require processing prior to human consumption because they have these poisons in them which cause men to go sterile, but that's a different story. It's a very important cash crop. It's even an important part of cash. Our dollar bills are about half cotton. We can't tell. The rest of it's mostly linen, but they won't tell you the exact formula, of course. That's a state secret. Okay, so tonight I'm going to introduce you to the cotton genus, which is the genus Gossypium. It's a story that involves the repeated parallel domestication on opposite sides of the world of wild species by humans, all for about the same reason. A remarkable story where evolution has recreated the same experiment, if you will. Humans have conducted an experiment over the last 7,000 years that we get to take advantage of today. They've replicated it 4 times. It's also a story that involves whole genome doubling. It's a process called polyploidy. You all have 2 sets of chromosomes, one from mom and one from dad, but what if you had 2 from mom and 2 from dad? Well, that's what a lot of plants do. In fact, it turns out that that's what all plants do. And I'll come to that a little later. So I'll talk mostly tonight about the history of the cotton genus and this mysterious process of polyploidy and what happens after 2 highly differentiated genomes growing on opposite sides of the world became slammed back together in the same nucleus at the time of hybridization and polyploid formation, and the mysteries that this generates that we're trying to solve. Now, I want to file a complaint with the dean that 25 years ago when I came to Iowa State University, I was given 45 minutes to give a talk. And here I am 25 years later, and I still have only 45 minutes to give a talk. So I can't cover very much of this. So what I'll do is I'll divide up this work about polyploidy and about gene expression and all this molecular biology into 3 short stories. And you can even sleep through 1 or 2 of them. I'll come back to them at the end, and hopefully you won't have missed anything. Okay, so here's a map. It's an old map of acreage. It's a very important plant. Cotton is a very important plant in the economy of the southern tier of the United States, as you can see. Fond of pointing out, it's very important in the economy of one lab in central Iowa, which you can see here. Very happy. That's a happy emoticon. You maybe can't see tossing dollar bills in the air. But cotton is a relatively recent immigrant to the United States. It just got here in the last several hundred years, you know, after white man invasion of the New World and subjugation of black man. In 1860, 60% of the population of South Carolina was actually slaves, the vestiges of which we're all, of course, living through today. Incredibly important plant in our sociology, economy, commerce, you name it. And from this New World location, the most popular species of cotton spread worldwide, forming the dominant fiber plant in the world today. You can see here in this slide it says, cotton, one of Egypt's greatest treasures. I didn't take this picture. I stole it out of a Land's End catalog. But what I like so much about it is that this is actually a New World plant that was domesticated by humans at about the same time as those buildings in the background were being built. This, of course, is a very common story in the annals of plant domestication. But it's a wonderful juxtaposition of the time involved and also evokes this, the role of humans in this germplasm exchange that's taken place recently in the last several hundred years. Okay, so David mentioned, and I'll mention the domestication process. The plant on your right is Gossypium barbadense in its modern, highly annualized form. You may have seen Sea Island cotton or Pima cotton if you've dropped a couple $100s on a fancy shirt or sweater. Luxury threads and yarns. About 5% of the world's cotton commerce comes from Pima cotton, shown on the right. It was domesticated, this high-yielding, annualized form bearing long, strong, fine white fiber was domesticated from tropical antecedents, shown on your left, that are photoperiod sensitive, bearing very sparse, short, tightly adherent, dingy-colored fiber, or lint. And this happened 4 times. 2 in the Old World, and those are diploids, like you and me, although we're paleopolyploid, but I'll come to that too. I am a paleopolyploid. I have a T-shirt that says that, actually. But 2 of them are also polyploids, and I'll come to that. So we have 4 independent domestications that each trace back, all involving these same morphological transformations. 2 in the Old World, 2 in the New, all going back about 5,000, 6,000, 7,000 years. The trait for which cotton is justifiably famous is shown in this slide. They're single-celled epidermal trichomes, as I mentioned. On the day the flower opens, this is what an immature seed looks like. And you can see that 1 out of every 3 or 4 cells is already terminally differentiated to become a single-celled fiber. 3 days later, they look like this. They've embarked on their nearly unidimensional growth. By 2 months later, that's 60 days post-anthesis, 60 days after the flower opens, they've formed these flattened, spiraling-like cells, these ribbons. The cell has died at that point. Over 95% cellulose. They're nearly pure cellulose. And the spiraling cells sort of flip-flop, and because of that, they'll stick together, they'll adhere, and they can be spun into yarns and threads, and that's what makes cotton such a wonderful fabric. But there's nothing like that in nature. I put this on a white background to show you what humans wrought from antecedents that look more like this. Wild species in the genus have seeds that variously look like this, or like this, or like this. These are species from Mexico, Africa, and so forth. The genus is worldwide, and what humans did is this kind of transformation 4 times for 4 different species, and it's shown there in the right-hand side of your slide. The genus is a medium-sized genus. There's about 50 species worldwide in warm, dry places. Here's Gossypium australe from Australia, and here's one from my colleague John Nason actually took in western Mexico. You see the cotton in this picture? No, Rob Wallace, that is not the cotton. This is right here. There's actually, I could have shown you a lot of pictures of cotton. There's 10-meter trees in western Mexico that are cotton. There's tremendous diversity. Here's my favorite clade, my favorite group, 17 species from tropical northwest Australia, this area that gets about 3 meters of rain a year. It all comes in 3 months, and then everything in the vegetation burns. And so you have all these fire-adapted species that send out these long vegetative runners. They have these beautiful mauve flowers that do a backflip after they've been pollinated right on ground level, and they release seeds that have now suppressed all their fiber, but instead they make a nice fat body that the ants run off with and plant. It's a wonderful story of an evolutionary extension of an intimate ant association that happens throughout the rest of the genus. Students often have trouble conceiving the evolution of complicated characters, complex traits. Well, here we have the antecedents still living. Throughout the genus, cotton, because of its extrafloral nectaries, the nectaries on the bottoms of the leaves, on the flower, they've hired ants as mercenaries to defend the plants. Well, now it's just a further continuation of that relationship. Now they're hiring the ants also, not just for defense, but to plant the seeds. Wonderful genus, fantastic diversity. I became very much in love with it. I wanted to understand it. And the way we do this in biology is we first construct phylogenetic trees. And now we do that using gene sequences primarily, and we've done this for thousands of genes for Gossypium, not the whole phylogeny, but we've sequenced thousands of genes for some of them. And the history of the genus looks like this. Ready? Boop. Of course, it didn't originate in Antarctica. I just don't know how to get it. I'm not good with PowerPoint. I don't know how to get it to come out of the screen at you, but that's what I'd like to do. So the genealogy of the genus is shown there in yellow. And what it gave rise to are groups of related species that we now designate with a single genome designator, a letter. So I'll refer to, for example, the D genome species that are primarily Mexican. There's 16 species. Or the K genome species that are from Australia. There's 17 species. Well, it's really nice to have a phylogeny. We can understand antecedent and descendant conditions better. It's especially nice if we can date it. We did this by sequencing 10,000 genes from the A genome and the D genome cottons. We know there's about 1% sequence divergence in the exons, for those of you who care, about 3% divergence in the introns and the UTRs. So we date this at about 5 or 10 million years ago that the genus originated. The continents were largely in the same place. So now we have an understanding of the relationships, when it happened, and now we can use this phylogeny and the dating in a powerful way. Provides a powerful framework for understanding the evolution of diversity. So we take, for example, their genomes. All of these diploids have the same chromosome complement, only 13 chromosomes, and yet their genome sizes vary enormously. They vary threefold, from about 900 megabases in the New World D genome diploids to 2,600 megabases in the K genome. They all have the same complement of genes, basically. Yeah, they all have the same genes. But their genomes vary threefold in size. What is all that other stuff? Why did evolution create it? What's it there for? What are the dynamics and the evolutionary processes that give rise to this variation? Complicated stories. One of the world's experts on this subject is sitting in the audience, Corinne Grover, who's studied this for many years now, and has shown that this largely reflects the differential amplification, differential proliferation of these jumping genes, these transposable elements, different families of them, as well as different rates of deletion in these different genomes. It's fascinating. We don't know the implications of all this, but we're elucidating the processes and the patterns right now. What else can you use phylogenies for? You can use them to study morphology. So here I've superimposed just the seed diversity in the genus onto the phylogeny of the genus, and you can see, for example, where long lint first evolved. It first evolved in the A genome diploid ancestors of what will turn out to be the New World allopolyploid cottons. Everything else here has short, non-spinnable lint. There's the ant-dispersed ones from Australia. So you can look at morphology. You can study the biogeography of the genus, knowing the date, only 5 or 10 million years ago, and the continents are largely where they are today. You can diagnose dispersals to the New World, an original long one about 5 or 10 million years ago, one that was probably just after that to Australia. Oh, cotton really gets around. There's one in Hawaii. You want to see that again? Right there. A recent dispersal to Hawaii, and I have to go look for that plant about every February. It's a wonderful time to leave. That's why I'm not in class then. There's one in the Galapagos Islands. There's one in Peru. And this is a wonderful mystery because the seeds sink like rocks. And so we don't really know how this happens. But it's important. Things do get around. In May 1990, 60,000 Nikes spilled from a storm-tossed cargo ship in the Northeast Pacific at about 0.1. 3 months later, they were found in northern Canada on the coast, and then sequentially down the coastline into northern California. By about 6 months later, a year and a half later, they were found in Hawaii, number 9. And in number 10, they made it back to, following ocean currents, all the way back to the islands off of Asia. And I understand from the internet that people were having swap meets and trying to find match pairs and having gatherings of the Nikes that were found. Similarly, in January of 1992, 11 steel containers spilled in the North Pacific near the dateline. 29,000 bath toys, duckies, turtles, froggies, and beavers were released. And the only reason I'm telling you this example is that I very rarely get to say duckies, turtles, froggies, and beavers. But by the next year, remarkably, some of the, well, let's just say the duckies, because they're so cute, made it through the North Atlantic Passage and were found on the coast of Maryland. So it's amazing what gets around, and I'm fond of pointing out that Hawaii has a flora and a fauna, and it's the most isolated oceanic archipelago in the world, and so cotton gets around too. This leads me to the final dispersal that I want to talk about, which is a really important one. And this happened in the mid-Pleistocene. We know this from molecular dating, about 1 or 2 million years ago. And that was another dispersal of the A genome to the New World. Only 1 million or 2 years ago. We don't know how. I've shown it here crossing the Atlantic. It could have been the Pacific. And this mystery was one of the mysteries that first attracted me to the genus some 30 years ago when I was still a graduate student. And maybe I should preface this. One of the benefits of getting older is you are no longer afraid of...
Where did it come from? 10 million years ago, scraggly cotton ancestors evolved separately on different continents.
So here's Mom. Mom is from Africa. Here's Dad. Dad is from Mexico. How did they manage to get together? How can you interbreed when you're in different continents? Well, Mom crossed the ocean. We don't know how. Floating on a natural raft, maybe on a bit of volcanic pumice. Who knows? Maybe a constipated duck got blown off course in a storm.
Mom and Dad got together and a whole new cotton was born.
Okay, so was that worth waiting for the technical trouble at the beginning? I'm not so sure, but this gave rise to a whole new lineage of plants, this process, irrespective of how it happened. And these are the allopolyploid cottons. And this is where the fun really began for me, because these allopolyploids, the AD genome, allopolyploids, now contain 2 genomes, an A genome and a D genome. And this is a wonderful mystery, because you like to think, let's just imagine that you have, well, let's not imagine, let's say you have 30,000 genes, okay, which you do. And you like to think of all those 30,000 genes having been finely honed by natural selection through the fires of the evolutionary process to work in beautiful 30-part harmony to guide growth and development and reproduction. So now I've just told you about these 2 genomes that evolved in isolation for 5 or 10 million years, each with their, in the case of cotton, 40,000 genes, each acquiring all their distinctive regulatory differences. And now imagine if Mike Galimo or if someone from music was in the audience, take 2 bands with 40,000 instruments singing a different song and put them back together. And how does it work? How does that guide growth and development and reproduction? And who's the composer? So this is the mystery of polyploidy. What happens when these 2 differentiated genomes are slammed back together in the same nucleus? In the case of cotton, it gave rise to a whole new lineage, and this is a very common process in plants. I wrote here it's ubiquitous. That means it's everywhere in flowering plants, and it's recurring. So botanists recognize different kinds of polyploids. That's not so important here. But if you make a tree of flowering plants and you put on little gray ellipses wherever there's been a genome doubling event, it looks like this if you only include a tiny fraction of the genome doubling events. This is such a common process in plants. So the history of plants is genome doubling followed by a shakeout, jettisoning a lot of that extra DNA, and then it happens again. Sometimes frequently, sometimes not so frequently, sometimes once every 100 million years, sometimes much more often, sometimes in our lifetimes. So it's ubiquitous, it's recurring, it characterizes 100% of angiosperms. And in fact, if you were to tally up all the genome doubling events that have taken place in the history of the genus Gossypium, I posted a figure like this on my door, and I've asked my graduate students to tell me the ploidy level of cotton. And it's not tetraploid, which would be 4 genomes, or octoploid. It's actually 48 ploid. And I had to figure out how to say that in Latin, and it's a real tongue twister. So ready to have your tongue twisted? Ooh, twisting? Duodequinquagintaploid. Everybody now. Duodequincogentiploid. That's 48 ploid. So that's the ploidy level of cotton, sort of ancestrally, but not now. Normal chromosome pairing is rapidly behaved. But this is a little bit of an epiphany for me because it helps me understand the origins of genome complexity in modern plants. You can now understand how plant genomes can have 100 of the same kind of gene. You don't have to even invoke special kinds of replication of the genes to get there. It's not just historically important, it's ongoing. In our lifetimes, there's new polyploids forming. In our parents' and our grandparents' lifetimes, there's some famous examples being studied by my colleagues around the world, mostly in Europe and in the United States, where these new species have originated in our lifetimes. We've watched them come to be. So this is evolution in action. And of course, there's neopolyploidy, or new polyploids in cotton as well, the ones that combine the A genome and the D genome, as shown in the lame video I showed you. And this is what they look like. In the upper left is the Hawaiian Island endemic. Then there's one in the upper right from northeast Brazil. There's one in the lower left from the Galapagos Islands with the lovely name Gossypium darwinii. And then there's the plant in the lower right, which looks like this. And this is Gossypium hirsutum. This is wild Gossypium hirsutum in its native form. That is, it's growing right there sort of in the salt spray zone in a coastal area. This is very typical of wild allopolyploids, growing in negative association with humans, a rangy, shrubby, photoperiod-sensitive plant with a very short, agronomically primitive seed covering of fiber. Seeds require scarification for germination, fully integrated in native vegetation. And this is the type of plant that first attracted Aboriginal domesticators some 5,000 or 6,000 years ago. This led to the development of all these dooryard and semi-domesticated forms that are really common in the tropics today. I took this picture in Colima. If you talk to the households, they maintain a few plants just out in the yards, and they'll collect the fibers for a variety of uses: lamp wicks, pillow stuffing, wound dressings, hygiene products, all kinds of uses, and they tend to flower irregularly, and then later, maybe thousands of years later, land races were developed, annualized forms were developed, and then photoperiod sensitivity was lost. It became a highly annualized form that now dominates world cotton commerce, as shown here in this 40-centimeter tall, storm-proof cotton from the high plains of Texas. From the New World, it spread worldwide to account for 95% of the global cotton commerce today, as shown here in this picture of this worker from the Ivory Coast resting on his work product, which nicely mimics a bowl of cotton. This is taken from the book The Earth from Above by Yann Arthus Bertrand, if you know that lovely book. Okay, pause to catch my breath. That's the organismal side of the story. That's your introduction to Gossypium, diversification, the origin of the polyploids. And it's repeated for you on the left. There's a summary diagram of what I just said. You have divergence from a common ancestor. You have independent evolution of two diploid genomes. And then they become reunited, this chance biological reunion, hybridization, genome doubling in the mid-Pleistocene 1 to 2 million years ago. And that translates into the genetic depiction on the right, or if you will, the expectation on the right, where the polyploid now has two of everything. And the question is, what happens when those two copies, those two homeologs, are brought together in the same nucleus? So that's what I've spent about the last 15 years on. And this is where the three stories come in. So the first story is that it turns out that Gregor Mendel has an evil twin, and this is what he looks like. And he has a very strange name, non-Mendel. Non-Mendel. And why do I say this? Well, here's your organismal history again, and that translates into the null hypothesis shown in the center of this slide. But we find, if we start walking through the genome, looking at bits of duplicated DNA, trying to see if they agreed with this hypothesis or if they falsified this hypothesis, we find all kinds of wild and crazy variations on this. So, for example, there's one called gene conversion, where instead of having two copies of a gene, we now have one, or kind of 1.5. One copy has overwritten the other copy, even though they're in different genomes in the same nucleus. So it looks like this. At the top you have the parents, an A genome and a D genome diploid. And if you look at the polyploid, they should have both copies, but they don't. One of them has been overwritten. In other words, this can be a diversity eliminating process. We also find genes that are chimeric copies between the two that were donated by the two diploid parents. This is a diversity generating process. It turns out there's a lot of these. It's about 3% of the genes in the genome. This is actually just being prepared for publication right now, exhibit this process of gene conversion between duplicated copies in the same nucleus. We don't know about the functional significance of this, but we're just describing the process. Here's another really dramatic example that you can see at the chromosomal level. You take the major ribosomal genes. There should be two kinds, one from the A genome parent, one from the D genome parent. Here what you have is you have a chromosome squash of polyploid cotton, and you see there's blue chromosomes and pink chromosomes. That's because it's painted with the pink parent, the D genome parent. So the small chromosomes are all from the D genome. You can see the pink ones are small. But irrespective of where the ribosomal DNA is, which is the big blobs the arrows point to, they all are D genome, even D genome copies. Even those that exist in the A genome, they've been overwritten. So you have this massive gene conversion process that's taken place. It's actually wiped out about 400,000 kilobases of DNA. Okay, that's one kind of non-Mendel. Here's another kind, unequal rates of evolution, this mysterious process that we're investigating further. Even though duplicated copies are in the same nucleus, one copy accumulates mutation much faster than the other. We don't know why. You can have transfer of sequences from one genome to the other. You can lose one gene and not the other. So these are all sort of non-Mendelian mechanisms, and that's story one. Okay, what's story two? Story two is algebraic, and I hope there's not any mathematicians in the audience. I see a statistician, so I'm already getting nervous. But this is why 1 plus 1 is not equal to 2, Ken, and why 1 plus 1 divided by 2 is not even equal to 1. And so it turns out that George Bush had a deep understanding of polyploidy, and that when it comes to gene expression, they don't follow additivity. This actually started about we started looking into this. Oh, what year is this? So it must be about 10 years ago. We wanted to know when two copies, if a genome all of a sudden has two copies of a gene instead of one, how are they used? How are they deployed? Are all of them still expressed? You know, you have DNA, then you have RNA, it's transcribed, so that's called expression at that level. Are they all expressed, or is there tissue-specific partitioning, or is it greater than the sum, or is it just an average? And what's the dynamics of all this? So over the years, we've done a lot of experiments, and we still are doing a lot of experiments trying to describe this phenomenon. Here, I'll just give you some take-home messages. So on the right-hand side of the slide, you see a polyploid triangle with the two diploids at the bottom and the polyploid at the top. And then you see three little percentages, and those are the percentages of differentially expressed genes. So that means that half the genes between the two diploids are differentially expressed. A and D. That's what the 53% means. Well, compare the polyploids to its two parents. Look at that asymmetry. In fact, gene expression in the polyploid there globally resembles that of one of its parent. It doesn't matter whether the D genome is upregulated or downregulated relative to the A genome. That's what the allopolyploid does. It's a wonderful and mysterious process, and Mijong Yu and Emmanuel Sidkowski, who are in the audience, are investigating this further in my lab right now. It's a wonderful, mysterious global phenomenon that characterizes the genome of polyploids, and it's now been found in a couple other polyploids as well. There's also a lot of up and down regulation beyond, so 1 plus 1 is not 2 here. It's a lot lower or a lot higher. This is transgressive expression. This happens immediately with genome merger and doubling. Evolution accelerates that process, so we've been studying that on a temporal scale. So globally, that's what happens. What about on a gene-by-gene level? So here we started looking at the two copies of a gene and how the plant is using them in different organs. So if you look at the top, you'll see all these organs. There's leaves, roots, stems, cotyledons, bracts, petals. And the top band in this electrophoretic gel that you see comes from one parent, the D parent, and the bottom band comes from the other parent. And if there was equal use of both, you'd see equal bands at the same intensity. Oh, and if you look to the far left in leaves, for example, it's not quite equal. The actual number is shown below. It's about 2/3, 1/3. But if you scan all the way over, you can look at the different parts of the same flower, they can use one copy in one part of the flower and the other copy in another part of the flower. So to the extent that both copies, that expression of this gene is essential in these floral whorls, both genes are then preserved, as it were, by this tissue-specific expression. This may have something to do with the significance of polyploidy on a long-term evolutionary scale. Well, if this happens in different whorls of the same flower, what about different parts of the same cell? So we started looking at fibers. And we looked at Gossypium hirsutum, which is the dominant plant of commerce, and Gossypium barbadense, which I told you was the Pima cotton, or the expensive cotton. And we looked at, and the arrows there show where they were originally domesticated in different parts of the Western Hemisphere. And it shows that they were both domesticated and gave rise to agronomically similar forms in Gossypium hirsutum and Gossypium barbadense. And we studied these during primary and secondary wall synthesis. And we looked at all the genes, and you find everything under the sun. So sometimes you find the genes, the A copy and the D copy, both expressed about the same throughout development. Sometimes you have only one copy expressed, and it just kind of grows. Sometimes you have equal expression at the beginning, but it transitions to all one copy. There's even genes we found where you have one of the duplicate copies expressed early in development of the cell, and the other copy expressed later in development of the same cell. So it's mechanistically really quite interesting and leaves me scratching my head. The take-home message here is that about a third of the genes change duplicate gene expression even in the level of a single cell. This has something to do with domestication because domestication accelerates this bias. And this is something else that's interesting, is that the plant is not using its two genomes equally. It's using the D genome more than the A genome in its fibers. There's a bias toward utilization of one of the two genomes. That's particularly interesting because of story 3, where if you look at the pictures in story 3, and I just told you it's using the D genome more than the A genome, it's really quite mysterious. There's the D genome, little crummy, non-spinnable, agronomically uninteresting, but beautiful wild plant that's much like most of the genus. Here's the A genome, which is where spinnable fiber first evolved. That's the African parent. If you look at the allopolyploid, it doesn't look average. It doesn't look additive between its two parents. It looks much better. So this is your other algebra lesson, is that 1 plus 1 is actually much, much greater than 2. Some of it has to do perhaps with the utilization, the excess utilization of genes from this agronomically inferior parent. So I'll remind you again that cotton domestication involved 4 different species, 2 at the diploid level, 2 at the allopolyploid level. And so we've studied the fibers of these, gene expression in the fibers, and the results look something like this. If you take fibers from 2 days after the flower opens to 25 days after the flower opens, and that's so 3 weeks, little 3.5 weeks of development, and you just, this is wild cotton over here, and you just sort of chart how many genes change expression during development, throughout primary and secondary wall synthesis, you can put in the numbers. And that gives you, okay, so I should explain what that means. So that 1,175 genes are upregulated at 2 days relative to 7, and 1,377 are upregulated at 7 days relative to 2. And you can look at the numbers, and you have these periods of relative stasis and these periods where the transcriptome, the gene expression, is really dynamic. And that's really interesting. Those are huge numbers of genes, and that's of interest to physiologists and cell biologists and molecular biologists and anybody who's interested in biofuels. This is telling us how a plant makes a cell wall. And this is a wild plant. So that's of interest, but it's not of evolutionary interest. That's just sort of developmental biology. Great interest there. You can do the same thing for domesticated cotton, and you put in the numbers, and you can see the same story, but the numbers are even bigger. So domestication has increased the dynamism of the gene expression program during cotton fiber development, more so than in the diploids by a long shot. This is one reason that 1 plus 1 is much greater than 2. Also, I just can't help but point out the crazy stasis in the middle of primary wall synthesis before transition to secondary wall synthesis, and then this extraordinary change in this 10-day period there. But again, that's just a developmental question. That's really of interest to cotton breeders. Tremendous information that one can mine from these data, how to improve and make a better fiber. But what an evolutionary biologist is interested in is that transition right there. And so if you put in the numbers for this transition, this is what it looks like. And if you total up all those numbers of genes that are differentially expressed, this represents the number of genes in the genome that were affected by humans as they practiced selection on wild cotton to make new cotton. That's huge. Some of the same genes show up in some of those categories, but that's over 9,000 genes. So humans affect in a single cell. So evolution is really complicated. Just selecting on this single cell over a 5,000 to 7,000 year period, humans affected the transcription, the gene expression of a quarter of the genes in the genome. So this caused me to change how I think about evolution. And I think, for me, a nice way to think about it is in terms of a big network. You know, think of your three-dimensional fish net and tug on it. But now make it 9,000 dimensional and tug on it. And so it goes something like this. So you have a cell, and the cell's doing everything a cell does. Transcription, translation, it's making a proteome and a metabolome, and there's all this feedback and all this regulation. And you can think of that all happening in this sort of network sense, and it guides, and it ends up leading to the construction of something that a human being might be able to see, called a cell wall, and that's how nature makes a wild fiber. And humans come along and they start practicing selection. So humans are tugging at the phenotype at one end. Well, there's not 9,000 mutations happening in those 5,000 years. It's probably, if we know from other crop models, it's probably, 5, 10, 20, okay, 50. 5 major mutations and a whole bunch of little ones, or maybe 50 little ones. But it's not 9,000. So those mutations are happening at the DNA level, and humans can't see that. This is 5,000 years ago. That's changing the shape of that network. So about 9,000 genes are changing their expression. That's how you get a modern cotton fiber out of that. So to dissect the shape of that gene network, what we're now doing is we're taking little bits of wild cotton, we're isolating them in cultivated cotton backgrounds, in near isogenic lines, and so we can plant all them out and study the great details, dissect the details of this, what we call genetic architecture. Okay, so that's one reason why 1 + 1 is a lot greater than 2. The final one I'll tell you about, and this is the end of story 3, I promise, evokes this image again, where the D genome contributed a rather short fiber to this cultivated plant, and yet we have this wonderful quote. Again, trying to bring in a little history here. If as a consequence of polyploidy, a large number of genes become duplicated, and the characters governed by such genes are of importance to the species, one of the members may mutate, leaving the character unimpaired, with the further possibility that the mutation may be of benefit to the species. So this is classic gene duplication or polyploidy theory. What it says is you've got 2 copies of every gene, so one's free to play around while one takes care of business. And so modern theory is that, yes, that's true, one's free to play around while the other takes care of business, but it's a race between mutational obliteration and getting a new job. So either you get a new and useful job or you're out of here, because a nucleotide substitution or deletion or something will happen and you're gone. Okay, that quote was 75 years ago by a famous old cotton geneticist, and I love the fact that we're now living in an era where we can actually see the details of this. So we're beginning to study this. We're mining the data set for genes that look like this, where the crummy-looking genome is donating to the only domesticated cotton. This is the signature of this get a new and useful job and its importance during agronomic improvement. And this is a complicated slide, so I'll explain it a little bit. Here you have 2 cultivated species of cotton, and here you have the diploid parents, and here you have levels of gene expression. And this slide is from Mi-Zhang Yu's work, and she's in the audience tonight. And you show here in green the level of expression for the A copy, in blue, the level of expression for the D copy, and you can see there's no expression anywhere except parallel under domestication. And we're finding that in a number of genes and mining the data set for more. So this is the signature of the parallel recruitment of D genome genes, and maybe helps us understand the importance of polyploidy and agronomic improvement. Okay, so that's pretty much what I wanted to say, and here's what I tried to say. Here's your top 10 list of evolution in polyploids. The first thing I tried to point out in the original part of my talk was that, and I should have given a little more homage to those who came before, They're really standing on the shoulders of generations of basic plant explorers, cytogeneticists, geneticists, agronomists, all kinds of scientists came before. We are able to harness the power of phylogenetics and molecular biology to provide really a rather exquisitely detailed insight into the evolutionary history of the genus, the origin of the allopolyploids, and the origin of the domesticated forms of the allopolyploids, and to put that in a temporal context that's justified with real data. And using that framework, we can now study how two differentiated genomes get along in the same nucleus. And we've learned a number of things. There's non-independent evolution of duplicated genomes. Gene expression in polyploids resembles one parent, a mechanistically mysterious and phenomenologically interesting problem that we're starting to explore in more detail. Bias and silencing of genes is ubiquitous. There's a lot of novel up and down regulation. We call that transgressive expression. You get expression partitioning even at the level of a single cell. I'd love to know how that works. The contribution of two genomes to a polyploid may be unequal. In fact, we don't even know that the two genomes are equally represented in the genome anymore. Gene conversion may be biased towards one genome or the other, or overall. Selection affects networks of thousands of genes, even at the level of a single cell. Duplicate genes may be responsive to selection. And polyploidy offers novel avenues for evolution and crop improvement, and will help us all develop muscle.
Open the window, shed all that steam, and let the wind write your name with every wave. The touch, the feel, the fabric of our lives. The touch, the feel of cotton, fabric of my life.
Kate Bosworth knows about polyploidy. Okay, so finally, I'd just like to thank, of course, all the people who really did the work, and they're listed here. Only a partial list of after 25 years, you can imagine the number of wonderful collaborators, students, postdocs who worked on one or more aspects of this problem. And of course, it's a huge team effort, and we live very much in an interdisciplinary era in science. And of course, my funding agencies, and finally, I'd like to thank Dean David Oliver for this opportunity to speak to you again, and all of you for coming out once more. Thank you very much.
Holding together long enough to ask a few questions?
Sure.
Okay. Well, yeah, why don't you go to the mic? I think we're big enough. Well, then let's see if the mic works.
Fred can boom.
Yeah. Bottom of it.
Yeah. Hi, Fred.
Talk about how the D genome can be differentially expressed in a lot of cases in running, and that has to do with the fact that even the D genome from their original polyploidization event came from just from the female, and because in the plants, the chloroplast, the other organelles might have a coevolutionary relationship to the nuclear genome. That sort of thing may be going on.
Yeah, that's a good question. He's asking about the cytonuclear dimension of polyploidy. One of the things I didn't point out, but that's really true, and was another one of the great mysteries, is that not only do these two differentiated genomes get together, but they do so in only one of the two parental cytoplasms. And it turns out, in this case, it's the A genome cytoplasm. So all the New World tetraploid cottons contain Old World plastid DNA, and all the New World cottons contain Old World mitochondrial DNA. And what's so much fun is that in my lab right now, Lei Gong, who I see in the audience, has started to look at rubisco, the most abundant protein on the planet. And the large subunit is encoded in the nucleus, and the large subunit is encoded in the plastid, and the small subunit encoded by a multi-gene family in the nucleus. And it turns out there's real signs of coevolution between those subcellular compartments. Good job. Any more questions? John? So I don't know if you all heard the question. The question is, why does the D genome dominate? Is there some section of the plant that the A genome would do better at? And maybe we should look there to see where the A genome dominates, perhaps. Yeah, I mean, the A genome does better in the fiber. In the D genome, there's a biased expression of the D genome. It's not huge, but there's a bias towards the D genome. There's even a bias towards the D genome in seed filling. Guanjing Hu, another, is Jing here? She's teaching. Another student in my lab. We recently looked at the way that the seeds, the duplicated genomes, contribute to the cotton seed because it's such an important meal seed. Remarkably, nobody even knew what the proteins were in cotton, one of the most important feeds on the planet, and nobody even knew what the major proteins were. It turns out that the allopolyploid cotton, the two genomes contribute unequally to seed filling, and the D genome predominates, even. Home in a geographic sense, yeah. 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 Geographically, or ecologically rather, the polyploids occupy kind of a new niche relative to the both diploid genomes. All the diploids in the genus are really sort of inland, arid to seasonally arid plants, whereas all the wild allopolyploids are littoral, and they're quite happy in salt spray zone, as you know from collecting tomentosum in Hawaii. Yeah.
Is there any evidence of elements from one genome, like transportation elements of things, jumping onto neighboring genome? Can you trace that with experimental process? Like
Yes.
polyploid?
Yes. Yes is the answer. Yes, there's evidence, and yes, we've seen it. Yeah. We don't know what it means. It'll be really fun when you find some functional significance, that kind of insertional event, where it's caused some phenotype that you really love. And you have a smoking gun at one end, and you have a phenotype. The smoking gun being some molecular biological event, and the phenotype being something that people care about. Yeah, then it's really exciting. Then you make the newspapers. We don't know. We don't have the question was, is there a compositional bias of where transposable elements, when they jump across genomes, seem to end up? We don't really know. We don't have a full genome sequence yet. We're about to get one for D genome cotton, maybe within a couple weeks. That should be, we'll be writing that up. The A genome soon to follow, we hope. We still don't have a polyploid, which is what we really need to get that, but that'll come shortly, I'm sure. Yeah, and the assembly problems and all that. Robert.
So you said that cotton was not originally evolved in Antarctica. Where did it evolve? Center of origin.
We don't know for sure. Excuse me. I got ice cube. We don't know for sure, but the center of diversity is certainly Africa. I wouldn't be surprised if that's its origin. From a formal cladistic standpoint, we don't know. It could, because the D genome seems to be sister to the rest of the genus, which has an African origin, but with an Australian extension. So my money's on Africa.
Did Vavilov say anything about his center of origin?
Vavilov, Saunders, all kinds of people put it in Africa as a center of origin, and that's largely based on the center diversity arguments. That's where most of the genome groups are, or a lot of them. Yeah. Kevin.
So this might be a simplistic question, but so have you constantly tried to recreate this experiment that happened sort of naturally? You recombine these parent genomes, you always get the same result. That one
So if you're asking the question about the bias and the dominance, we don't know enough to generalize, although we've seen that we get different genomes dominating in different polyploid processes. Is that good enough? Okay. Thank you very much.
Jonathan, one more time.